May I have your attention? We will be starting with this program. A very good morning to everyone. I am Queenie Torin Nongrum, Research Officer under CSCMI, and I will be comparing for this program. On behalf of the Indian Institute of Public Health Shillong and the Center for the Study of Complex Malaria in India, it is my great pleasure to welcome each and everyone to this stakeholder engagement meeting. A special welcome to Dr. Biba R. Marak, State Program Officer, NVBDCP, Government of Meghalaya, Dr. Marba Warshong, District Malaria Officer, East Khasi Hills, Dr. M. Solia, Medical Officer, NVBDCP, and to all our special dignitaries and to my fellow colleagues. Now, as a token of welcome, I would like to call upon my fellow colleagues to present flowers to some of our special guests. I request Mr. Peter J. Marbanyang to present the flower to Dr. Biba R. Marak, State Program Officer, NVBDCP. I request Mr. Bandap Kupar Kliang to present the flower to Dr. M. Warsham, DMO East Khasi Hills. I request Ms. Matimi Pasa to present the flower to Dr. M. Solia, MO, NVBDCP. Ms. Inam Kentio Lingdo, can you present the flower to Shri Larry Karbaman, State Monitoring and Evaluation Consultant, NVBDCP? Mr. Bayene Nongri, can you present the flower to Professor Sandra Albert, Director of Indian Institute of Public Health? Thank you. I now call upon the Director, Indian Institute of Public Health Shillong, and Principal Investigator of the Center for the Study of Complex Malaria in India, Professor Sandra Albert, to kindly give the introductory remarks.
Good morning. Am I audible? Okay. Yeah. Right. Can we have the first slide, please? Right. The Center for the Study of Complex Malaria in India, or CSCMI, involves a national and international collaboration of researchers from multiple organizations. On behalf of our collaborators, in particular, Professor Jane Carlton, the Program Director, I welcome you all. Dr. Biba Marak, SPO, other representatives of the Health Department, and all special invitees, we are delighted that you could join us today. Our Principal Secretary is delayed at a long meeting with the DC. He has sent his representative, Kong Alvarine, to join us today, and thank you for coming. He will probably be joining us a little later and has asked us to continue. Next slide. The CSCMI is one of the 11 centers for excellence in malaria research, that is, the IICMR initiative funded by the National Institute of Health, USA. Capacity building of young researchers and transfer of technology are key components of this initiative. Next slide. Professor Jane Carlton from New York University set up the CSCMI over a decade ago in India. She started with Chennai, Gujarat, and Orissa as the study sites. In 2016, she reached out to me as malaria was endemic to Meghalaya, but not much research was coming out from this area, and hence it was almost like a black box. Thus, during the second round of the IICMRs in 2017, we put in a proposal. Meghalaya was brought into the picture to take up epidemiology work, with a focus on understanding what the asymptomatic status in malaria infection really meant. There is a lot of work going on in Odisha as well. For example, Dr. Sanjeev Mohanty, Sam Wasserman, and their team are doing cutting-edge work to understand the pathogenesis of cerebral malaria in Orissa. They have MRIs set up and a lot of intensive work is going on there. For example, I just want to tell you an implication. A standard treatment regime in cerebral malaria was to give mannitol to reduce cerebral edema in falciparum malaria. But after their studies, we now know that this approach could potentially lead to increase in morbidity and mortality for these patients. So we know now lots more about pathogenesis of cerebral malaria. But I won't go into that. That's for another day, and we can share papers which are coming out from their research with you if you'd like to know more. Next slide, please. When we started planning work in Meghalaya, we had to rely on the 2016 figures for malaria in Meghalaya. The areas in red on the map of Meghalaya correspond to high prevalence districts at the time. This was what guided us in site selection and sampling for our epidemiology work. Next. We started first off with an analysis of data from the NVBDCP's decadal data from 2006 to 2017. On the graph, the number of cases are shown on the x-axis to the left, and mortality is shown to the right. There was certainly a reduction in malaria since the distribution of over 941,000 LLINs in 2016. But if you observe the graph, you will notice that the numbers of malaria cases had already started falling prior to the LLIN distribution. Hence, it is possible that these effects resulted from a combination of factors, possibly the other control measures that are implemented, or even other factors such as health system strengthening initiatives post-NRHM that were taking place in our rural areas. Next slide. Since 2018, we have been undertaking different studies in Meghalaya to assess baseline malaria epidemiology, prevalence of PV, PF, and asymptomatic infections, identification of recurrence of Vivax infections, and risk factors that potentially impact malaria transmission. And you will be hearing more about all this later today. Next one. In September 2019, to enable molecular level diagnostics that require testing that goes beyond rapid diagnostic tests, a malaria research laboratory was set up and formally inaugurated by the Honorable Chief Minister and the Honorable Health Minister. And again, you will hear more about this later. Next. One area of malaria that is relatively neglected in our country is entomology, or the study of vectors, in this case, the Anopheles mosquitoes. So we made a small start under Kathy Walton's guidance. Normally, morphological ID is used, i.e. by looking at the wing and leg patterns of mosquitoes. But if you look at the wing and leg patterns that are shown there, they belong to two different species. But you can see that this can be quite challenging even to the expert eye. Hence, molecular ID can help in such instances for actual and accurate identification. Next, please. Although we recognized it was important to work in the Garo Hills, due to safety concerns in 2018, we deferred work in this area. But by 2019, you can see on the map that South Garo Hills alone remains the brick red area, showing high API or annual parasite index. So we started work in the Garo Hills in 2020, and then COVID hit. But with the support of the MD&HM and you all, we have been able to resume work in this very difficult terrain. You may have seen some of those images that were flashing on before we started about some of the really challenging situations that my team had to deal with when we were doing when we do work in South Garo Hills. Next. We are also doing some work in the area of socio-behavioral studies, a novel method that we are attempting in collaboration with Dr. Eliza Bellotti is social network analysis. In brief, we are trying to understand why people choose to do certain actions, who they talk to or avoid talking to, and so on. This is ongoing work, so hopefully when we meet next time for such a meeting, you will hear more about it. Next. And finally, Dr. Melissa Glenda put together the slide for me. I should also give a shout out to Mr. Frankie Starr and Dr. Eva and Mr. Tingshan from NLHMB, who work behind the scenes with bills, auditors, paperwork, and admin work, which really allows us and gives us time to get on with the research. But yes, coming back to the slide, recall, I said the center was about capacity building in the state and the region. I'm pleased to report that we have been able to employ over 47 people in the past 3.5 years. Over 91% of them have been from the Northeast region, especially Meghalaya. The spread of the personnel from the different districts in Meghalaya is shown on the map. And 94% of our employees belong to scheduled tribes. We have therefore kept our promise. Today, you will hear a few of them make presentations to you. Most of them from our MC, who you've already started to hear from, to the last closer of the session with a word of thanks, are addressing an audience such as you for the very first time. So bear with us and do encourage them. Thank you very much.
Thank you, ma'am. Now we will move on to the presentations of the activities in the field and the preliminary findings, which will be presented by the three teams. Firstly, I will call upon Ms. Phibansuk Lingdo, Field Coordinator for the Epidemiological Study of Malaria, to give the presentation.
Good morning, sir, madam, and everyone. Thank you very much indeed for coming today. I am Phibansuk Lingdo, the Epidemiological Field Coordinator of the Center for the Study of Complex Malaria in India. I'll present on the fieldwork related to the epidemiological component of the study. Next slide, please. So to begin with, I would like to give you an overview on the ongoing epidemiological studies in Meghalaya. So we have the clinic studies in 2 districts, that is the West Khasi Hills and South Garo Hills, the Plasmodium Vivax cohort study in West Jaintia Hills, and the community survey in South Garo Hills. A description on each study component will be briefed in the upcoming slides. Next slide. So this is a schematic diagram of the epidemiological study workflow. It is a very large study. However, I will talk on the section circled in blue. One of my colleagues, Mr. Bandab, will brief on the other sections. The diagram depicts the data and sample collection process. So sample collection proceeds in the following order. That is the microvet, the rapid diagnostic test, the blood smear, and the blood spot. Next slide. The clinic studies. The clinic studies came into force on 2018 at West Khasi Hills, that is at Nonglang PHC. It was then extended to West Jaintia Hills and South Garo Hills on 2019 and 2020. The ongoing clinic studies are at 2 sites, that is at West Jaintia Hills and South Garo Hills, Narting PHC at West Jaintia Hills and Siju PHC at South Garo Hills. So basically, only individuals presenting to the PHC area with malaria symptoms are referred for enrollment in the clinic study. In particular, the ones advised by the medical officer to undergo the malaria parasite test. So the prevalence of Plasmodium infection identified in the clinic in 2020 is 4.7% in the West Jaintia Hills and 7.4% in the South Garo Hills. In West Jaintia Hills, as opposed to the South Garo Hills, we notice a mixed species, but not in the South Garo Hills. Next slide. Another ongoing study at West Giant Hills, NIRTH-ICMR is the Plasmodium Vivax cohort study. The Plasmodium Vivax recurrence cohort was initiated after an outbreak at Mukbu village. This study was started on the requisition of the state NVBDCP. So the aims of the study, one is to assess the recurrence of P. vivax after unsupervised treatment, to evaluate the factors associated with recurrence, and to collect well-characterized series of recurrent P. vivax infection. This is to understand if the infection is either due to relapse, recurrence, or recrudescence. So participants identified as PV positive from clinics as well as a village are recruited and followed monthly until 2 years of follow-up is complete. So in 2020, 126 individuals were followed up, out of which 11 were detected positives. In 2021, 137 individuals were followed up and 3 were detected Plasmodium positive. Next slide. The community study. The community-based study at South Garo Hills District, CGPHC, was started on May 2021. The aims of the study are to estimate malaria prevalence, the overall malaria prevalence, asymptomatic, subpatent, to identify associated risk factors, age, gender, travel, anti-malarials. So comparatively, CGPHC represents a higher number of malaria cases in the past few years than other PHCs in the district. Hence, CGPHC was selected for the community survey. A purposive sampling method was adopted to select villages on the CGPHC, meaning villages with higher malaria cases in 2020 are selected or prioritized for sampling. A house-to-house enrollment is done. All members of the household aged 1 to 69 years are enrolled in the study, and participation is completely voluntary. Next slide. Now, I am going to talk about the task the team actually execute before, during, and after the field activities. So before the start of an activity at any village, the team would make a preparatory visit to the village to seek permission from the headman, to survey the village, and to sensitize the villagers. So sensitization is done by conducting a few meetings with a few representatives of the village, requesting the headman to make announcement to the general population of the team's visit, and visiting the usher and speaking to a few people in the village. During data collection, that is tasked during the activity. So during data collection, post-introduction, a team member explains the purpose of the visit and the study to the potential participants. Once consented to participate, 2 sets of questionnaires are filled. One is the household level questionnaire and the other is the individual questionnaire. So the household level questionnaire gathers information on the household size, the age distribution, the socio-economic status, the use of malaria preventions. And the individual questionnaire obtains demographic information like the age, gender, education, occupation, the travel history over the past 2 years, and the use of malaria preventions like the ITNs and the repellents. Anthropometric measurements are then taken. Height is measured using a stadiometer, weight using a weighing machine, the mid-upper arm circumference using a myotape, and the temperature a digital thermometer. So the instruments used in the field are displayed towards your right and will be explained by my colleague. Please feel free to interact with them, with our team members. Following the anthropometric measurements, the sample collection begins. So here is a picture showing sample collection being done during the night time. The time awaits on the convenient time of availability of the individuals. So the samples collected are 2 to 300 of microliter of blood is collected in a microvet for the PCR analysis. Falsivax kit and ultrasensitive kit is used for rapid diagnostic tests. Blood slides for microscopic detection. The blood spots for DNA verification or identification. And the HemoQ is used for checking the HP level of an individual. So after the activity, after a day of enrollment, the team then later prepares for the next visit. This table represents the number of residents from villages and the number of households and residents that were enrolled in the community survey. That is from May to September 2021. From the following 7 villages in this time frame, we have enrolled a total of 215 households and 755 individuals. Out of 755 individuals enrolled, 17 asymptomatic persons were detected Plasmodium positive, 5 that is from CG Duramang, 1 is from Nokut Kray, 2 is from Jadi Gindem, 1 is from Rongsu Agal, and 8 from Rongri Gitam village. The molecular identification are awaiting. From a past experience, more positives were identified through molecular identification, so this number may increase. To highlight on a few things of what went well and what are the challenges the team faced during the field activities. So what went well? The coordination and support of the District Malaria Officer, South Garo Hills, the Medical Officer, CGPHC, and the District Consultant, South Garo Hills, has been of great help in executing the study at South Garo Hills. The team has been in close contact with them to spot out the villages where malaria cases are popping up. On the other hand, the team also informs them if any Plasmodium positives are being detected at any villages in the district, in the villages that are under the CGPHC. A sincere gratitude to the monitoring and evaluation consultant, Mr. Larry Carboman, who has been constantly supporting the team throughout the course of the study. The utmost thing that made the enrollment process much easier for the team was the involvement of the ushers of the villages. The ushers' help was invaluable in the initial introduction for the recruitment process. Another point is the cooperation of the participants. The participants recruited in the study were very cooperative and patient enough, even if the collection process takes a longer time. Next slide. The challenges. Talking about the challenges, one of the main challenges was the road that leads to many of the villages is not accessible by neither two-wheeler nor four-wheelers. The team had to walk for 1 to 3 hours to reach the village. Traveling to and for the village was not possible at all for the team due to the poor road condition. Hence, the team had to stay over at the villages. Heavy downpour of rain hindered the workflow of activities. The prioritized villages that are very difficult to reach, such as Kemdang, Hansapal, and Dogring, where cases are being detected, could not be sampled due to this reason. Scattered household is another challenge. The households were very scattered from one another, therefore more time was consumed to reach the households. So convincing the individuals to participate in the study was another challenge for the team. COVID added to the challenge to convincing the individuals. However, there are other reasons. For instance, in some villages, the people were more concerned about receiving treatment for other conditions rather than getting themselves tested for malaria. There are also high rates of refusals in some area. So a few methods were adopted to reduce the refusal number, such as seeking the help of a key opinion person in the village, as well as showing the individuals the collection process up to the method of sample and analysis via a video and pictures. Somehow that has helped us reduce the number of refusals in some of the villages. So individual availability at the time of visit was also very high. The reasons for unavailability are migration and schooling. With that, I conclude my presentation. Thank you very much. If you have any question, I am happy to answer.
Just one question. Do you have any sample size for this particular study?
Sample size.
Rajeev, you want to take that question? Sample size.
Sample size, I think for the West Jaintia and the West Khasi Hills was about 2,000. For South Garo Hills, we have already covered more than 500. What we didn't, the challenge with this kind of a sample size calculation is number one, the prevalence is very low. So the sample size will become huge as compared to the population. So what we decided was to focus on the populations where there was more malaria cases with the hope that we'll be able to pick it up. Because I think the more than estimation of prevalence, because as you know, ma'am, the malaria is very seasonal. So if the prevalence, it will be very much dependent on what time of the year I'm doing the sampling. For example, if we do it, let's say around May or June, we may not get, or let's say April, we may not get many cases as compared to doing it in, let's say, September, when the cases will go up, especially in those areas. So that was one of the issues. So what we decided is we'll go with, that's what Fibon was mentioning. that we'll go with villages where there is active cases so that we pick up more cases. Because the interest from the South border was more than prevalent, because I think the incidence data will come from the IDA, the NVDA ECP data itself. But our main interest was what are the risk factors? What makes some people more vulnerable whereas others no? And in my presentation, I'll touch on some of the analysis that we did for West Jaintia and the West Khasi Hills, where we're looking at the risk factors in a low endemic area. Because the thing is, the vectors are still there. The environmental conditions haven't changed. So there is always a chance for resurgence if people bring in malaria in these areas. So, and that has been the story, I think, for malaria, from what I understand, for a long time.
So just one more question. I just want to know the importance of this anthropometric measurement in this study. What is the importance of that?
No, so I guess what we wanted to, we are looking at risk factors. So we kind of want to know whether any of that, because we know there's some amount of malnutrition, we know anemia, for instance. We want to know if there is any causal factors that can be arrived at. We haven't looked at the data in depth. We hope to do that, even though field work will come to a close by the end of this year. Yes, they just to know that. Yeah.
And I think anthropometrics are also doing it as a routine kind of an anthropometry for the people. So we'll also have some baseline on that. Also, Kiban didn't mention we are doing hemoglobin tests in this population.
So I've mentioned, sir, we're using the HemoQ.
Yeah.
So the instrument is there to check the HB level of an individual. HemoQ is used.
Hemoglobin, there is a relationship with malaria.
And malaria,
Correct.
yes.
In fact, we have found quite a few of them.
Yes.
Anemic, yeah.
Yes,
We
especially
find
in
quite
Sarkar
high
Hills.
numbers.
Okay, thank
I
you
think
so
one of the
much.
reasons for anthropometry would also be to link with malnutrition. If there is a relationship between malnutrition and malaria and things like that. In general, there is a relationship with malnutrition and infectious diseases. I don't think that it is established for malaria.
Thank you.
I'll just pick your brains a little bit. Just go back to previous slides towards the beginning, please. Yeah, just here. Okay. So these follow-ups, you have to follow them over 2 years, right?
Yes.
So 137 is inclusive of 126, like
Yes.
the second year.
Yes. Yes.
So you have more. in the
Yes,
second
we have more. Yeah.
year. And any issues faced? I mean, 2 years is a long time to follow up.
Yeah.
Lots of issues.
Yes.
So you have pointed out some, no, the
Yeah, I've pointed out.
migration
Yes,
and
this might-
studies.
That is in
and-
Sarkar, but here in the PV, people, one reason is people they don't want to give the blood every month. So that is one reason, which is why we work in collaboration with the SW, the surveillance worker, and the ASHA of the village. So we work along with them. So that is one reason, but we can't even force them to give their blood sample if they're not willing. So, but we never. So, if in case if any individuals they refuse more than twice, we do not visit them again. But again, we visit them in the next year or then after 3-4 months. Especially in the malaria season, we do visit everyone. So we do.
So one of the reasons was that although they have explained to them why we are doing it and it's to understand the relapse patterns, for like in winter months they just simply refuse it because they know there's no malaria in winter. They're like, so why do you want a malaria test during winter? That was one of the key issues for a lot of people
that is
refusing. But if you go back to them another 3 months later when it is malaria season, then they are a little bit more. Some of them are a little bit more.
They do participate.
So this has been a very challenging piece of work, but longitudinal studies are like that.
Yeah, and how would you then differentiate between a relapse and a recurrence?
Yes, so that is where the genomics come in. So there is no way clinically to do that. So that is why the genomics, and because of COVID, we have not been able to complete this bit. Some of the samples have now just gone to New York University in the United States, and so watch this space. Hopefully next time when we meet, we should be able to give you some
It's
idea.
been 2 years.
Yes.
Yes.
Because we couldn't even ship the samples. So we sent some of the samples, they had to be returned back from Guwahati because blood samples are not allowed outside the country during COVID. So we had all these huge challenges. But finally, I think there was one courier, the World Courier, who are hugely expensive to send this small parcel. It costs us, I think, over $1 lakh to parcel that. Yeah, that's the kind of money is involved. And we have sent it in because we realized we do need to get some results going forward and can't wait for so long. Yeah.
Thank you.
What do you do with these patients? You mentioned that there are some patients who come to the PHC, but the villages you could not visit because
Sorry,
of-
ma'am,
yeah.
come again.
you track the patients, you
So we go to the
identify
village.
the patients at the health facility.
No, ma'am. Actually, one is yes, we identify them at the health facility. They come to the clinic, we sample them, enroll them in the clinic. If they're tested positive, we also follow up those individuals. But also there were around 126 that during an outbreak at Mukubu, there were like a lot as reported by the NVBDSP. So we also recruited those bunch of individuals into the study because they were once tested PV positive. So we recruited them as well. So this was done in requisition of the state NVBDSP during an outbreak at Mukubu village. Yes, Mukubu village, ma'am.
So what about the ones where you said there were some villages that were inaccessible? So you
That
let
is in
those-
South Garo Hills,
You
ma'am.
let those cases go.
They were not included in the study anymore.
So that is in South Garo Hills. So we are waiting. Right now we're waiting. We'll see if the rain calms down. If the team can go to the village, we will definitely sample
South
those
Garo
villages.
Hills is also only PF, no?
Yeah.
Yes,
only-
yes,
yeah.
yes.
So they don't have to be followed up. They just need to be
Just
met
once.
once to take the sample for genetic studies. The point is, sometimes they go and they have to come back after all that, carrying all this material simply because they just cannot cross
The river. We have to cross the
once
Simpson.
Once the bridge was broken, right?
I think Dr. Trillian would know better. Yeah. So that is why. But then we're keeping those villages like Kiamdang, Hansapal, and Dogring at South Garo Hills on hold because cases were there in the month of June and July. So we're keeping them on hold. So once the team are able to go to the village, we will definitely sample those villages, ma'am. Yes. Thank you. Thank you so much.
Thank you, Pipan. If there are any other questions, we will be glad to answer them during the question and answer session later. Next, I will call upon Mr. Fornes Lamin, Field Coordinator, to give the presentation on the entomological study.
Good morning, everyone. My name is Fornes Lamin Amdab. On behalf of the entomology team, I would like to present the entomological work that we have done so far. So here the work, here the flow chart that we have to follow before commencing any activities in the village. First of all, we have to seek permission from the headman. And after we getting the permission or the consent, we approach the houses for the household consent. And here we also explain them about our work. And after explain them our work, if they like to participate with us, and we ask them questionnaire in related to mosquitoes. Next one. Okay. So here is sorry, I'm a little bit nervous because this is my first time. So this is how we collect sample. We collect mosquito sample by using light trap. It's a portable sampling device attached with a UV light for attracting the mosquitoes, vectors, and also a fan that keeps the catch in a collection chamber or in collection bags. So the trap are set outside of the house or mainly in the veranda in the evening around before 6 p.m. And the traps or the sample were collected in the next morning at around 6 a.m. So in one day, we deploy 8 light traps, 4 light traps for the household with a negative malaria, and 4 household for the household with malaria positive test. So this one is under sampling method that we use in collection of mosquitoes by sucking air through the hollow tube. So these are 2 types, the manual aspiration and the backpack aspirator. You can see out there, the first one, the figure 1 is a manual aspirator, and the second one is a backpack aspirator. So we use both of these method to collect mosquito samples in the cattle shed during the night. So after collecting the samples, we process them by following these steps. In the first step, we segregate all the mosquitoes, all the Anopheles mosquitoes from other mosquitoes and insects. And after segregate them, we identify them with the help of microscope. And then we label them by giving them the names, the collection number, the village names, and also the ID number to each sample. And after we label them, we keep the record, and then we preserve them in the silica gel for the subsequent molecular work. So we are not collecting only the adults, but also we collect the larva. So for larva sample, we collect by using the larva dipper. So we cannot see there. Actually, my friend's holding it, a bowl with the handles. So you can see out there. I keep it there. So we usually do this one in the daytime from the rice fields. From the rice field, stream margin, graupels, and also other water bodies, other freshwater bodies. And the purpose of this one is to find out the breeding habitats of the particular species, and also the potential vectors that present in a particular area or in the village. So same thing here, the larva collection, the sample processing for larva. So for larva also we did the same as we did for the adults, processing, but the difference is here only is that we rear them until they emerge. But once they emerge, we follow the same processing protocol like we follow for the adult samples. Next, please. So now we come up to the identification of the sample. So as you can see here in the slide, during the year 2018, our identifications, our identification is completely mismatched with the molecular ID. But however, in the year 2020, through training and our hard work, we have improved a lot in our identification. So these are the samples that we have been collected so far. We collected in 14 villages, 10 from the West Khasi Hill, and 4 villages from the West Gentia Hill. And total number of adult mosquitoes, we collected 2,409, and out of this, 18 species were identified. And 178 larva samples were collected till date. And altogether, the samples is 2,587. So this is the end of my slide. Thank you so much.
Thank you, Fornes. If there are any questions.
Yeah, what was the previous to your last slide from that? Before this, yeah, this, yeah.
Yes. Actually, ma'am, this is identification, our identification. This one we did by ourselves. This one is a molecular identification. and this one is molecular identification, which is done in the lab at PCR.
If I may just
It's
morphological ID, which they do,
which you
With
just
yes,
compare
manually with on the microscope.
And so this was not corresponding to the
Yes,
molecular.
it's not matched to this one. But if you see here during the year 2020, whatever we identify here is almost same with this one, except in some species which is very closely related. So like Philippines and Newipis is only a little bit different. Actually, everything is same, but only different is only in the wings, only small point actually. And also with the new species that is not there in our books. That's why actually it is in the same group, but then they are different when we come to species level.
So out of all these species that you got now,
Yes.
are they all vectors, disease carriers?
So the next presentation is going to tell you a little bit more about that.
So just want to know, ma'am, any specific reason for this discrepancy in the morphological identity?
Okay, we're going to talk about that after you, because we also did a pilot with the NVDCP entomologists collecting, and we did some of this molecular ID field. So maybe we'll take that once you've seen that, we can have a chat about that.
Yes.
Yeah. Thank you, Furnace. Well done. Yeah.
What's the data set for molecular?
So that's a good question. Again, you're going to hear all that. That's fine. It's okay for you. You don't have to take molecular ID questions. That's bad now.
We just received news that Dr. E. Shadab from Pasteur Institute Laumali has joined us. Welcome, ma'am. I request Mr. Fornes Lamin to please present her with the flower.
Thank you, ma'am. I'll be quite all right here.
Thank you. Moving on, I call upon Mr. Bandapkupar Maukleen, Research Fellow, to present on the molecular diagnosis of malaria parasite and associated vectors.
Thank you, Queenie. Good morning, everyone. Our colleagues and invitees from NVDCP. My name is Bandapkupar Maukleen, lab in charge of the Molecular Biology Laboratory at Indian Institute of Public Health. So today I'm going to present a very brief, presentation about the molecular diagnosis of malaria parasite and associate vectors and how the work is being carried out in the lab. We have heard from my colleague Piban of how samples were collected and preliminary processing was done at the field. So what do we actually do with all the blood samples that were collected in the lab? So from the small blood volume collected, DNA was extracted from the RBC samples for PCR detection, and the plasma was also preserved for immunological assays. From all the positive individuals whose 5 to 10 ml of blood was taken, we filter the blood samples and extract the DNA for parasite whole genomic sequencing, the RNA for gene expression studies, and the plasma for serological assays. The buffer code is also always preserved for human DNA and RNA extraction, and this is mainly for understanding the cell and mediated immunity involved in the positive individuals. This is the workflow that we follow in the lab for conducting PCR. So in brief, a PCR is a technique for amplification of a specific region of a DNA of any species. Firstly, the DNA was extracted, and because we use a very large number of samples, we could not follow the manual method, but we used the commercial QIAGEN kit. And then the extracted DNA was further concentrated in a speedvac machine. This is from Thermo Scientific. The concentrated DNA is used as a template for the PCR reaction, and the amplified sample were run in a gel electrophoresis, and we usually do it at 10% because the fragment size is very small. Gel electrophoresis, a method to separate the DNA fragments into their specific molecular size. So how do we visualize the results? For visualization of the PCR result, we use a UV trans illuminator. So here we can see the P Vivax positive samples, and these are from Nartiang, and those P falciparum positive samples from South Garo Hills. So for all the amplifications that we do, we always compare with against a standard positive control, there PV and PF. These are well-characterized sequences that we obtain from authorized repositories. So till date, the total number of PCR assays conducted are 1,753 from West Khasi Hills, 4,324 from West Jante Hills, and 550 from South Garo Hills. The total number of positive samples is 106, out of which 29 are both RDT and microscopy negative. These includes the samples from both the community and the clinic samples. So all the positive samples, we use them for whole genomic sequencing. The aim of this study is to identify and isolate which a distinct type of a species that pertains to a particular region. Also, we want to analyze the extent of genetic diversity that are present in these isolates, and to mainly to identify the P. falciparum drug resistance and the P. vivac recurrence. So this is an ongoing work that is being carried out at the sequencing facility at New York University, and I hope in few years we will have a very concrete results from here. So next I'm going to talk about the molecular identification of Anopheles species. Anopheline mosquitoes occur typically as groups of closely related species that cannot be easily distinguishable using morphological characters alone. Therefore, at CSCMI, we ensure that correct identification is done, and we do this by using a multiplex PCR method that detects the species belonging to Hircanus group, and other primers, we detect the species belong to Maculatus group. So what about the other species? We use the COI sequencing facility, and this facility we are using, you can see there in the far right at ICMR NIRTH. So what are the key findings from our molecular identification assays that were conducted? So 18 Anopheles species were identified till 2020. These potential vectors, many of which are broadly zooanthropophilic, they are found in diverse habitats and specifically forest fringe and rice-growing environments. Species diversity is observed more in West Janta Hills as compared to West Khasi Hills. There are predominance of very rare species like Anopheles japorensis. These are very rare species in the northeast. It's a predominant vector in Bangladesh, and also Anopheles maculatus. Two species considered to be predominant primary vectors in the region, Anopheles dirus, were not identified in any of the samples from West Khasi Hills and West Janta Hills, but we have just collected, I think from past 2 months, 2 samples of dirus from Garo Hills. Also, only one sample of Anopheles minimus was collected from West Janta Hills. A very important, let's say, a first report that is in India, we report 2 new species in India. This is Anopheles zui and Anopheles dissidens. In the context of changing malaria epidemiology, that is due to deforestation, changing climatic conditions, and also increased use of LLINs, Further studies are needed to determine which species are now contributing to malaria, and also to characterize their biological attributes relevant to vector control, including biting time, host preference, larval ecology, and also seasonal abundance. In 2018, we conducted a pilot study from the samples that we received from NVDSP from South Garo Hills and West Khasi Hills. So you can see in the x-axis are the samples that were identified morphologically. The species that were identified as Anopheles culisfacies, dirus, and minimus were actually not supported by our DNA sequencing results. Rather, they were identified by other species like Nivipes, Anopheles pallidus, Anopheles peditineatus, and Anopheles pilensis. So similarly, with samples that we receive from districts like West Khasi Hills, we have found such say misidentification, like it's also happening with our team. So we understand that morphological identification and tough and sometimes may be complicated by difficult to interpret taxonomic keys. Therefore, it is necessary to adopt molecular tools to ensure correct identification. And yes, like Fornes pointed out, we will all continue to learn, discover, and improve our understanding. Thank you all, the entire team involved in fieldwork, and also for the data analysis.
Can I ask a question? Yes. So, if they're not really that different, pharmacology and molecular, I mean, so they would be different in some aspect. Right?
Yes. Yes.
So now, that kept in mind DDT resistance by these vectors. Is that being studied?
Which is what we want to do. The only thing is when you want to do drug resistance or anti-vector resistant to the vectors, you have to have a certain number of them to do these tests. So we are all set for doing it. The unfortunate thing is our numbers of mosquitoes are insufficient because we have to grow them and have at least 50 in a cage to test certain things. So this has been a little bit challenge. So that is why we are now switching to try and do some of it through the known vectors and there is a certain genetic marking when they have developed resistance. We are trying to see if that is a way to it's a conundrum. We'd like to do this, and maybe in the future we could perhaps, but we haven't cracked how to get the mosquitoes and grow them and insufficient numbers for doing those kind of studies. Yes.
What you said was they're being so identical. So we do have species that are called cryptic species. They belong to different species, but they're morphologically they look the same. Or we have sibling species where morphologically they look the same, but they are subspecies within the same species.
But they behave differently. All of them.
So if a species belongs to a particular group, the behavior also is like almost same, which as compared to other species.
Almost first if you see this the anomalous. So anomalous actually we thought only one type of anomalous, but then when we come to molecular ID, we have anomalous A, and anomalous B.
Yeah, these sibling species, what we call.
But with our naked eye or with the help of microscope, we cannot identify that except with this molecular.
So yeah, I'm afraid the difference really lie at the at the molecular level.
Because our entomologists they're collecting a lot of mosquitoes.
Yes.
I don't know how we can coordinate them.
And Bandap, I just want to ask, you mentioned regarding the sample provided by NVDSP, it was in South Korea, especially it's all most of the species are there as well. Now you're saying a bit different. The one you receive from them,
yes,
not necessarily direct.
Yes. Yes. Yeah, similarly with samples we receive from West Khasi as well.
Is there a system where we can get this checked regularly?
So I think from the last stakeholder meeting, we also requested that some of these samples be sent to us, at least if not all, but a pool of them. But I think I think yet to receive them, or I think will receive in the near future.
One more question. Like, if they identify as non-virus, not virus also, are they a potential vector?
Yes.
Yes. Yes, true. Yeah.
So how many out of all these species that you have identified are actually vectors for malaria?
So that is what we are still doing. We are still sequencing. One sequencing for one fragment is doing at NIRTH and one is at Manchester. So yes, we'll come up with the.
What ma'am is asking is, so for instance, Japorensis, okay, it's hardly reported in India because we thought it's not there. But in Bangladesh, it's one of the key vectors, and we have identified it here. So it is potentially it is a vector.
Yeah, it
so
was.
Like that,
Yes,
like
ma'am.
that, some of the others also in other parts of the world, it has been documented as a vector. Here, no, we have. You can conclusively say something is a vector only if you collect the mosquito and there's blood in it and it is the life cycle is documented. That part we are not doing because we haven't. It's very difficult to get those blood-filled mosquitoes at that particular stage where we can. Yeah, that is the challenge. I mean, we'd love to do it.
200 you'll get.
Yeah, exactly.
Yes. So yeah, even.
Vectors also, ma'am, there in Bangladesh because they are primary vectors, but then in Meghalaya it's not considered as primary vector, but we found vectors in Garo here. Because it's close to Bangladesh.
One more. What about these analyses? Where do you collect them from?
The two species are not the new species. Where do you collect them from?
From West End Hillside.
Which area?
Zoe.
Which area of honors?
Anopheles zoe and dissidents.
These two species we collected from Baratu.
Baratu.
We collect it in a cartel shape.
Both the species from Bharat.
Yeah.
In fact, founders will tell you that it is much easier to collect from the cattle shells. But when they do in the homes, they have spent hours trying to collect in the homes. Because there are so many zero collection.
Because like 90% of mosquito you can find in the cattle shell. Even it's not feeding there, but there is rest there.
No, it's not that. We don't know what it is, whether it is because of the LLIs now in the house. We don't know. But the point is we are not being able to get them in the, or if it is new house. So the villagers use a lot of other methods like smoking, burning of the egg trays, all kinds of things. So we don't know if those have affected, and it's a good thing if mosquitoes are not inside the house. But it doesn't mean that they have to be biting you only inside the house, right? Biting can happen when you're sitting on the veranda and having tea or going out in the forest. And increasingly, that is what, Rajeev will be telling you something about the risk factors that we see, and you'll realize the relevance is actually not so much in the house now, it looks like it's outside the house.
Okay. Madam, regarding this concentration of vectors, I would say
Yes.
that most of the villages, there are 1 or 2 houses that will stay outskirts of the villages, nearly, especially near the forest areas.
Yeah.
Now, the malaria cases that we're detecting is particularly from the cattle houses.
Yes.
So if it's like, if you go to the entomological work, you have to consider those cattle houses which lay like outskirts of the village.
So they are the team in the field. So what he's saying is that you focus on the houses which are at the border of the forest and the village, right? Those are the places they are finding the cases.
Actually, sir, according to our study, we have to spread all the trap in the village, not only in the, in the forest.
But you can actively do that in the forest fringe too, forest. So that's a good suggestion. I think you should incorporate it.
Like direct, so you will not get you will get mostly in the forest.
Yes, ma'am. Very less. Even from the book that Dr. Lingdo wrote, for the past few years, we very less divers.
In fact, they have been doing an active surveillance for divers because Kathy Walton is particularly interested in this species.
Yeah, we want to do the population genetic studies.
But you got only one virus you had done last year only. Yeah, you got in 2
from Garo Hills.
One from Rongra and one from C. And even, sir, And because if he's not there, if he's already retired also, but we still contact with him.
Yeah.
So he said that before, yes, a lot of errors he saw, but then nowadays he said, I didn't saw any errors nowadays, he said like that. And
Any
he
reason for
said
that?
only the process, and, I mean, it's there, but then it's not like before. I mean, the quantity population. So even us also, we see the same.
One last question. Now, with all this data available, how do we translate this? We can relate this to our field stuff, like what we should be doing, we should not be doing.
That is
And
like
that is why we'd like to have this conversation with you.
Yes. So really, we do want some, samples from the entomology, from entomology. We have been, Valerie, we have been asking, so we could really like them.
You want it on a monthly?
Yes. So
let's try and do it.
so my question is, when they collect the samples and then when they identify and document it, what do they do with the samples? Do they preserve them or do they?
We dissect them. We dissect them.
Like we dissect them?
Yeah,
all?
we dissect them to find out if there's a parasite present or not.
So
Because we're not sure which vector is available. So we dissect all of them, make sure that at least if they carry a parasite, we could at least identify them.
So even in areas where there are negative cases, you dissect them?
All of them, all of them.
So can you at least give just, some samples which you don't dissect to us, like some samples?
Yeah, the thing is that, like I said, we dissect all of them. Because we're not sure.
But we get a lot of larvae.
Yeah, we can, larvae, but they don't care.
No, also if you get 10 of the same, what you think is all diurus, for instance, dissect 8 and send 2 to us.
What if it's 2 I send you guys is positive?
I know, but you have to take a call. Basically, that's why we are kind of between you all decide whether you want us to do it or.
It's not about being that. It's just
Can we not have larvae?
The larvae will not.
Because we get plenty of larvae, we don't get that many mosquitoes.
But larvae won't
You can
carry
always
the
give us
parasite.
larvae. You can always give. No, we can extract the DNA from the larvae as well.
Yeah.
We can do that.
Because you are rearing them anyway.
Yeah.
Yes.
Even if you rear the larvae, but
the larvae and they
The
bring
survival is
and
less.
yeah, survival is
larvae.
till it becomes adult mosquito. That also can be done, I think.
Yeah,
Yes.
I mean, rearing them is nothing. It's like just keeping in a basin with a little net on top. So if you can do it and rear it and then put it into to the silica gel and give it also.
What if fondness accompanies these boys?
that'll be better if they come. That is the you collect the mosquito, fine. Because even us we need them, no? That's the thing.
Yes, we can work.
Yeah,
it out.
if
We
you
can
can
work
do
it
that,
out.
that'll be
Yes,
fine.
sure. Thank you so much.
So you all have become quite the expert at identifying the species.
So basically, yes, I mean, fondness have improved a lot. I mean, when we compare the morphological ID and the molecular ID.
Yeah, I can see the difference between the 2 years.
Yes.
So what if our entomologists also bring some mosquitoes and then go through them together with you guys?
Yeah, I mean, that will be good because I'll tell you what, when we went for training Funes and team, first we bought entomologists from NIRTH, that's double O, because in India they are the best team there. But what we realized is they are very excellent at the North Indian mosquitoes. They didn't know that much about our ones here. So that is why they have to teach themselves in some sense. They attend a lot of, they're using the molecular ID key and or rather the morphological ID key, and they have been teaching themselves. Then when each time they get
the molecular
like Vasna and Kathy from Manchester, when they have done some of the molecular stuff, then they debrief with them, kind of to compare what went wrong, and so they review what they did. They're taking pictures, so all kinds of things. They're doing all kinds of additional things to build up their knowledge bank in some sense. Yeah. So we could develop something like that, so which will help your team. And for that we need to develop this bank of mosquitoes which are fairly common here, and then I guess we could develop some key with tips which these people have figured along the way. Yeah. We should talk about it suddenly.
Yeah, it's still very important to get the morphological identification right. Because first thing is that doing molecular identification is very expensive. It's very expensive. It's very expensive. And also, even if we do it, at least if it's identified correctly at the group level, so we can separate the PCRs rather than do everything at one shot and try everything.
Them to just identify the group level, because otherwise the kits are wasting really expensive reagents, doing it 3-4 times rather than getting it. So that's why we are persisting with them to some level of morphological ID being correct, and it's nice to know that they are.
Just to put them in the group is easy for them. I mean, but then when it comes to species levels, lots of species can be very tough.
Okay, thank you, Bandap, for the presentation. Lastly, I call upon Ms. Matimi Pasa, Field Coordinator, to present on the Social Behavioral Science Study.
Good afternoon, everyone. I am going to talk about the social and behavioral study to understand barriers to current malaria intervention strategies in Meghalaya. The aims of our study are to observe on how preventive measures such as long-lasting insecticidal nets and indoor residual spraying are implemented in practice. This aim has 3 objectives. One was to observe on the LLIN distribution. The second was to observe on how IRS is being applied, and this was done through non-participant observations where we accompanied the NVBD-CP team during deployment of bed nets and application of IRS. The third objective was to understand LLIN utilization at the household level, where in each village we visited up to 5 households selected randomly from each locality. Our second aim was to get an improved understanding on knowledge, attitude and practices within community and health system on malaria and its prevention. The technique used was focus group discussions and in-depth interviews. Our third aim is to undertake social network analysis to map social network of influence in a village to identify potential behavior change approaches that could support a malaria elimination strategy. The method used was interviews consisting of open as well as close-ended questions. On the observation of LLIN distribution, the table shows the LLIN distribution coverage in the selected study villages. We observe distribution to over 2,000 households involving over 8,000 LLINs distributed to over 16,000 community members. Overall, it was found that LLIN distribution in 24 of the 5 selected community centers achieved the intended coverage of 99.5%. On LLIN utilization observation, it was found that post-distribution of LLIN in 2020, utilization of pet nets compared to the villages we observe in the previous year has improved, and 30 of the 60 households reported that they have started carrying bed nets to their agricultural field. With regards to IRS acceptance, our team observed of 1,079 occupied households that were visited by the spray team, 58.6% refused to allow any spraying to be done in their homes. Only 18.4% agreed to be sprayed. That too, 14% were partly sprayed, and 4.3% were fully sprayed. The reasons for refusals are no adults were present during the visit. Villagers shared that there was not enough time to rearrange their household items. Presence of young children below 2 years was also one of the reasons for refusal, and the others were stated. We have a lot more, but then we presented few here. So, but it should also be noted that one of the reasons for refusal was that community members were worried if their beekeeping or airy silk moth cultivation would be threatened.
You actually had people telling you that they've never had IRS done previously?
Yes, ma'am. So in a few of the villages in West Jaintia Hills, so they said that the reason they refuse is that even before they have never done it, they haven't accepted.
IRS has been there in their village,
yes, but they are not. Yeah, they won't
accept.
accept.
It's not like IRS.
Oh no, it's not that.
We have never done it till now, so I do.
Yeah, that's why they don't want. Moving on to a second aim, which is to get an improved understanding on knowledge, attitude and practices within the community and health system on malaria and its prevention. Here, in-depth interviews with health personnel at the village level, including the MO, ANM, laboratory technician, pharmacist, GNM, ASHA, community health volunteer and traditional healer was done. We also conduct focus group discussions with the community members to get their perception. We have conducted 72 FGDs, which includes 616 participants and 55 in-depth interviews in 19 villages from West Khasi, West Jaintia and South Garo Hills respectively, and the data is under analysis. But I will brief a bit on how we process with the analysis. So we are using thematic content analysis or the framework analytical approach to interpret our findings. In the thematic analysis, this is a method used for identifying, analyzing and reporting patterns or themes within the data. The framework analysis is an inherently a comparative form of thematic analysis which employs an organized structure of the derived themes, that is the framework, to conduct cross-sectional analysis using a combination of data description and abstraction. So the steps that we followed in a thematic analytical approach are first, we get that is familiarization of the data. We first read and reread the transcribed data. We then generate codes to identify important features of the data relevant to answering our topic of discussion or research question. In the screenshot to the right, the comment box, I'm not sure whether you can see it. Okay, so okay, the comment box represents the codes. So after coding, we then arrange the generated codes into themes and sub-themes. The fourth step, we then, in this phase, the themes are refined, which sometimes involve them being split, combined or discarded. The fifth step, that is defining and naming themes, in this step we finalize and describe on an informative name for each theme. The last step, that is the write-up. This involves weaving together the analytical narrative and data extracts and contextualizing the analysis in relation to existing literature. So the third aim, this aim uses social network analysis to map the structure of social influences within a village. In each village, we interviewed every adult included in the ASHA health registry to whoever who consented to be a part of our study. One-to-one interview was done and the respondents were asked to name up to 5 people they talked to or discuss and avoid talking to for health-related matters or malaria-related, 5 people whose opinion or expertise they go to for health or malaria-related matters. We also record the rate of adoption of malaria preventive measures such as IRS, LLIN, wearing long sleeves and full-length clothing, insecticide creams, coils, etc. Our next step is to document and understand health system factors. Here we will conduct key informant interviews with senior officers for expert opinions on implementation of the program. With that, I conclude my presentation. Our acknowledgement goes to our collaborators, the SPO, the consultants at the district level, and of course my enthusiastic colleagues for their hard work, for without them our field activities would not be successful. Thank you.
From the studies that you have done so far?
Ma'am, for the observation studies, I have presented it here already in these. Also, we have shared a manuscript with you in the folder titled Implementation of the...
Oh, this is the one,
yes,
the detail.
So it's only the qualitative part which we have not been presented because we are still doing the analysis. And the third aim, so the analysis is being done by Dr. Eliza Bilotti from the Manchester University.
So for that we don't have any results as yet? Not yet.
The third, no,
Next
ma'am,
time.
not yet.
We have some, we have been discussing it, there's some early results, interesting things to be shared with you next time. Yeah, I think that will be very important even with vaccine hesitancy. We realize it has wider implication.
Thank you.
Thank you, Matimi, and to all my colleagues for the presentations. I now would like to call upon Dr. Rajeev Sarkar, Project Coordinator, CSCMI, to present the analysis of the epidemiological data.
Thank you, Queenie. It's afternoon, so good afternoon, everyone. Esteemed guests from the NBDCP in the Health Department, and students and dear colleagues from IIPHS Shillong. I'll talk a bit about the analysis of some of the epidemiological data that we did and some of the findings from there, titled as Epidemiology of Malaria in Low Transmission Settings. As Professor Sandra and others have already mentioned, that we have done different studies, and my focus is going to be the community-based studies in West Khasi and West Jaintia Hills, which was done in 2018-2019 in Barato and Nonglang PHC areas, and this is the data that I'll be. The analysis of this data is what I'll be presenting. I mean, South Garo Hills fieldwork is still ongoing, and hopefully in our next presentation we'll have some findings from the South Garo Hills too. As far as the survey is concerned, we did this study in about 22 villages in the 2 districts, West Jaintia and West Khasi Hills, covering about 1/3 of the total population and half of the households in 2018. We found very low prevalence of malaria during our cross-sectional survey. So what we did in 2019 is we kind of curtailed down our sample size and the study. So 2019 we covered much less of the households, even though we were planning to cover more and to focus on other studies. So most of the results will be for the 2018 survey. of the about more than 2,000 people whom we surveyed and from whom we did get blood samples, we got 33 positives, and 97% of those 33 positives were asymptomatic. That is detected only by PCR. They were RDT negative and microscopy negative. And that was one of the reasons why Fiban, when she was presenting her results for the West Garo Hills, did mention that we are still to do the microscopy, the PCR analysis of those samples. We may get more positives from the South Garo Hills collections.
This is from South Garo Hills?
No, this 33 of 2,000 plus, that is about 1% prevalence, is from West Jaintia and West Khasi Hills in the year 2018. So this is an interesting observation. We just wanted to know, where our samples are coming from in relation to the population distribution. So this is the 2011 census population of those two districts, West Jaintia and West Khasi, and at the bottom is what we sampled in our survey, and we can very clearly see that our participants were more likely to be young children or women, which is probably what is expected because of migration and other things that happens, and this is probably true of all programs. So there is always a sample there's always a bias in terms of the sampling. So there's always a yes, that's the age, and yeah, that's the population pyramid actually for the sample population and the study population. So just for us to keep in mind when we look at the results, because to kind of put it in the context. So as I was mentioning that the prevalence, we found 33 positives, so the prevalence was about 1.2% overall, and as I was mentioning that 97% of infections were asymptomatic. Of course, the prevalence didn't vary a lot between West Jaintia and West Khasi, with West Khasi Hills at that time in 2018 being a bit higher at a prevalence of about 1.3%. Whereas West Jaintia was about 1.09% or 1.1% roughly. This was an analysis of data, and this has been published. I think it's one of the handouts in the folder. This has been published, which we look at, where the NBBDCP data was analyzed over the years, and it was seen that there was a good number of cases reported in 2015, 2016, 2014, 2015, but from 2016 onwards the cases started declining, and as we know that the malaria cases are very few nowadays throughout the Meghalaya. I'm not going to elaborate on this. We also did an analysis, a logistic regression analysis of the different risk factors, and we did it two ways. We looked at the village level risk factors using a simple logistic regression, and essentially we took village as the unit of the analysis. And what the risk factors that came out at the village level was presence of electricity, presence of animals and mosquitoes. They were all risks. But we also did a more complex analysis where we used a multi-level model to basically look at the individual and the household level risk factors. Within that model, it's basically a nested. And what we found is that in the individual level, we found that the history of malaria in the last 12 months, and in the last 12 months in the travel history, a recent travel history, where independent risk factors or factors associated with malaria at the individual level. So this is the individual level risk factor. We have also put it into a paper which is under review currently, and we have also put it in your handout, so you can have a look at that also. And it was a detailed analysis, but these were the only two factors that came out of the regression analysis. We also looked at the spatial clustering, because we were all in an area where there was malaria historically, and we know that the vectors are present and the environmental conditions are conducive. We expected that there could be pockets of transmission, and how to identify the pockets of transmission. We used something which is called as a SAT scan analysis, which basically we plot the GIS coordinates of the villages and the number of cases in each of these villages, and what it does is it runs a circular window. So it starts off with a larger circular window, and then it kind of makes it smaller and smaller to look for clustering, where it will look at, and in comparison to the other areas around, where it will look at what is the observed number of cases reported and what is the expected, and that is where the statistic comes from, which is a relative risk. And we did find a cluster in the West Khasi Hills area for the 2018 data, again in the southeast of the Nonglang PHC area, where we found 11 cases reported in those villages, as against an expected of 2.15 which is in the SAT scan. And that is also something that we felt was this is an important good methodology in terms of looking for pockets of transmission if we want to look at the data overall. We used a Poisson model because we used the number of cases as an outcome variable, but there is other models that can be used. For example, there's a Bernoulli model, which basically looks at presence or absence. So there are different models that can be used. It's a software which is freely available, so it's also not something that is expensive. So and looking at the data in that way, we found for not only for malaria, I think we have also looked for other diseases where we have found this to be helpful. We also did some serological analysis using 17 antigens against Plasmodium vivax and Plasmodium falciparum, 13 Plasmodium falciparum and 4 Plasmodium vivax, looking at lifetime exposure. And as expected, what we found is that the exposure, the percentage of people who are seropositive for almost all these antigens increased with age, because there was, they would have gotten exposed at some point in time. But significantly, we also found that there were exposure in the children. Here, the under 5 children also, we found some exposure, which kind of, again, looking from the asymptomatic, looking at the data for the asymptomatic transmission, strengthens that there is some underlying transmission that is happening, and we need to be careful about that. There are studies in very low transmission settings where people have found that there is no positivity by RDT or by the microscopy, but there is serological evidence that is there. So even longitudinal serology can give us some idea about how the transmission patterns are over time. Of course, we did an analysis. This was a data which we collected in terms of the risk factors for malaria and what are the preventive methods. And as I think Mati has presented, and it is good to know that her data agrees with the data that was collected from the cross-sectional study, where we found that almost everybody used an ITN, everybody used a LLIN in this area, which probably explains the low transmission. Also, we found that there was a less than 50%, I think, used IRS or allowed IRS. So the IRS coverage was low, again, as exactly what Mati had found for pretty much the same reasons that she has outlined. So I'm not going to go elaborate on that. To conclude, the NBB DCP data clearly shows that the malaria incidence has declined in the study villages between 2014 and 2018. And this is again what we found also in the 2018 survey, where we found a little more than 1% prevalence of malaria in our cross-sectional studies. We did found majority of infections to be asymptomatic, which is there is a potential for low intensity transmission in this area, because the RDT or the microscopy would probably miss some of those cases. And also we need to do more studies, of course, to look at the relative contribution of asymptomatic, because when we did 2019 survey, which I did not present here, we also found that some of the people who were positive in the 2018 survey, all of them were negative in the 2019 survey without any treatment. So what does it mean in terms of the program if the person is having a submicroscopic infection, if the person is present? One option could be to actually look at gametocytes to see if there are viable gametocytes, which are the ones that would be transmitted, which we did not do in this particular study. So that is a potential limitation. I guess we can think about that to really kind of find out what is the role of this, because there are studies where they have found that asymptomatic or submicroscopic transmission could contribute to the ongoing transmission in an area. So that is one of the potential areas of further exploration which we think we could move into. Of course, given there is a spatial clustering which identifies pockets of transmission, and also the fact that a recent travel history was a risk factor at the individual level, there is a potential for resurgence. And we know that we are in an area where there are malaria endemic regions and countries surrounding us. For example, Bangladesh, Myanmar are both endemic areas. So there is always a chance of resurgence through travel and other things, and we need to be careful about it when we look at the malaria epidemiology in a declining malaria context. So we need to be a bit vigilant in that front. And there are again examples where this has happened, where there was no malaria and a lot of malaria was imported. So that is something. And of course, because of this low levels and the fact that a lot of our, even the cross-sectional survey positives were RDT negative, we think that we could think about conducting periodic serological surveys to look at the longitudinal transmission pattern. So if we can do repeated cross-sectional surveys, then and maybe monitoring the under 5 kids and under 5 children, we can also, they could give us an evidence of recent transmission, because their lifetime exposure history would be much smaller than an adult population. So thinking about that, and of course we did discuss about insecticide resistance, because that is something that we need to consider, given that a large proportion of the decline probably can be attributed to LLIN, and we know that there is parathoid resistance which is being reported now globally. So that is something that also we need to keep in mind to ensure that we keep low levels of transmission, at least from our initial findings. That is where I'll stop. I'd really like to thank the CSCMI team. I think Fiban, Bandap, Fonas, Mati, and all were here. Peter is here. You heard from some of them. They have done tremendous work in some of the pictures that Fiban had shown under really, really difficult circumstances. Also like to thank Dr. Aprup Das from ICMR NIRTH and our collaborators from the New York University, from the University of Manchester. Special thanks to Professor Mark Wilson, who really has been really guiding us in terms of this data analysis. He has been working quite diligently with us on that, so thanks to him. And before I conclude, just wanted to highlight the fact that although our malaria work through the CSCMI project will be winding up, the fieldwork will be winding up this year, and while we'll have a few more years to complete our data analysis and reporting and other things, I'm quite excited to kind of inform you that we will be starting with a new project, which is a Zoonotic and Vector-Borne Disease Research and Training Center. And from our discussions, ma'am, what we learned is that this is probably also going to be covered by NVBDCP. So we will come back to you with more discussions on that, where we'll be covering 6 diseases: scrub typhus, brucellosis, Japanese encephalitis, cryptosporidium infections, H1N1 influenza, and leptospirosis in this project. And hopefully this will also help us develop. Professor Sandra mentioned about the field staff distribution and in terms of capacity strengthening for projects, but this project will help us strengthening of capacity, not only in terms of the field personnel, but also more importantly from the lab capacity building. And we are quite excited about this project. This has been funded by the DBT Wellcome Trust India Alliance, which is part funded by the Department of Biotechnology. It's kind of a not-for-profit organization, part funded by Department of Biotechnology and the Wellcome Trust UK. So here I'm going to stop, and I'd be happy to take more questions. Thank you very much.
So just one question from my side. Regarding the study, the epidemiology of malaria indoor transmission settings, basically it is a cross-sectional study. But then I just want to know the sample selection. How did you select those asymptomatic cases of malaria? The asymptomatic samples.
So basically what was done, and Fiban can elaborate on this because she is the one who actually went out in the villages to recruit. So we went to a village, we did a selection of the households in the village, about half for larger villages, and we basically went door to door, asking that if they would be willing to participate, and those who agreed to participate and provided a sample, we collected the sample. The sample was analyzed by microscopy, by RDT, as well as by PCR. So each sample that was collected was analyzed 3 ways.
You were saying that, Rajeev, that the asymptomatics of 1 year on follow-up were found without treatment to have no parasites again the next year.
That is correct. So we did look at it. There was a small number of people. See, our positives are only 33, as I said, so it is a very small number. And I think we were able to follow up around 17 of the 33 on the next year, and found none of them were positive.
Yeah. And none of them received treatment.
None of them has received treatment.
None were positive. Sir, have you done anything for these asymptomatic cases which you have detected by
I think that has been a debate, and I invite Professor Sandra to elaborate on that.
So this is in the community, malaria research community, this is a big debate, and the answer is still out there. But I'm just going to use, and we don't know what it will be the story in Garo Hills, but if you look at the West Khasi Hills, okay, so we detected, say he said 33, about 1% of positives for asymptomatics. But when we followed them up in 1 year later, they were all negative. In other words, their body was able to deal with it and it became spontaneously regressed. So why subject someone
study or something?
Yeah, which is what we are kind of doing only because of the limitations of travel. We have done some, and he presented some of the antibody studies. Many of them seem to have antibodies already. and that's probably what's working. We do know that if there was spontaneous resolution in a majority of our cases, in West Coast Hills definitely. So I guess in some, I think the answer policy decision, because it's a big decision, right? It's better probably not to be hasty and demand treatment at this moment. I think it's well worth the wait.
I think there could be different things to it. For example, PCR, and from what I understand from Banda's presentation, they are basically doing an amplification PCR, so they're doing some sort of an amplification. So it could detect very small number of DNAs, parasitic DNAs. That is one thing. Now whether how
so the concentration can be till 20 nanogram. Okay. Okay.
So
so not significant to have a parasitemia for transmission to carry on.
Sometimes the parasite is very low. It's very low. So it cannot be detected by slide.
So but that's a good question to ask. Does transmission happen? That is the key question, isn't it?
They don't pose a threat as a carrier.
They do. That's the thing.
No, I think if I may,
they can.
Treatment should be given.
From what I understand, I think the transmission part of it. The transmission part of it, I think the one bit that we have not looked at actually is do they have viable gametocytes? Because that is what will define the transmission. Now we know that they have parasites in their body, but what form of the parasites is one question. And of course,
Gametocyte for transmission to
yes, exactly. And the second thing is that what is the number or level of parasite that is needed for parasitemia or clinical symptoms to manifest, given that in the adult population, majority would have had some antibodies because of repeated exposures. Because the antibody data also shows that about almost 80 or 80% of our adults greater than 18 years had been exposed. There is an exposure. There is an antibody to the antigen that they have seen. So I think this will be an important study probably to look at within the low transmission settings, and also to kind of answer the question about do they need to be treated or not. Because if it is very low, if there is very negligible gametocytes present, which is the risk of onward transmission is quite low, and they can clear it out by themselves, do we still need to treat them? Because detection would also require yes.
Can I say something? Yeah. So you must remember that PCR detects genetic material.
Yes,
It doesn't detect the whole
Yes.
thing. Okay. Same thing for COVID also, what we're doing right now.
Yeah.
We're detecting the genetic material, but we do not know the virulence of it. So the same thing can be used for this, for explaining this. They could not detect it by RDT, could not detect it by microscopy, they detect it by PCR. PCR detects genetic materials, small scrap also. Remember, it's amplified. So very small amount also. So you may be detecting only the dead genetic or degrading genetic material also. That's why the debate you're talking about treatment carries on.
I think the gametocyte is the thing.
Yeah, absolutely. And that is one thing which we couldn't do because of all the other limitations. Finding that.
If we find a gametocyte, then we treat. If we don't find a gametocyte, then we can let it go.
I think.
I cannot answer that.
And I think in terms of epidemiologically, to detect if there is an ongoing transmission, possibly cross-sectional serological studies looking at a young age group would be something that will be helpful, because children, if they are born, they would not, unless they are exposed, they would not have that antibodies. That is what, there would be some maternal antibody transfer, but that should be cleared within the 1 year or so. So if we look at, let's say, 1 year to 5 year kids, we may be able to answer some of those questions in terms of longitudinal transmission happening or not happening. Excellent. So thank you very much for your patience hearing, and thank you.
Thank you. Thank you, sir. Next, I would like to request the State Program Officer and VPDCP, Dr. Biba Armara, to give some remarks on the works of the CSCMI.
Good afternoon, everyone. Dr. Sandra Ma'am, Director of IIPH Shillong, and the whole of IIPH team Shillong, my colleagues, the state consultants, and the district consultants, and all the NVVDCP team present here, and everyone who is present here. First of all, I would like to thank Sandra Ma'am for giving me this time and opportunity to stand here and give my comments on the excellent works that you all have done in the field. So first of all, I would like to congratulate you all, everyone, the IIPH team and the Center for Complex Malaria, India. So this meeting, the stakeholders meeting, is really a very it's a meeting that we have gained a lot from this meeting. So I'm very glad that our team, the NVVDCP team, is here, sharing our views and giving our comments. So before going into whatever comments we have to give, it's already been done, I think. We have already discussed along with the presentations. But just going to tell you all in short about the NVVDCP program. We all know that malaria is declining. Keeping 2015 as the baseline, in 2020, we have come down to 96% of malaria morbidity, and by 94% we have come down in terms of mortality also. But even in terms of API, the major indicator for malaria morbidity, it has come down to less than 1. So in spite of all this reduction, we still have got some few hotspots in some few districts like South Garo Hills, in Jaintia Hills, and also in some parts of West Khasi Hills. So just in short, I want to just tell you how we have come to this decline of malaria in Meghalaya. So, like we all know, Meghalaya is endemic for malaria because of its peculiar climate, the topography, the terrain, and we have already discussed the communications. So this also leads to a huge diversity in vector distribution. We have already discussed on that also. So just highlighting the key interventions that we have been implementing in Meghalaya to bring malaria to such a decline within these last 5-6 years. The first tool for surveillance, that is the early case detection and complete treatment. So here we have engaged our ASHAs. We have already trained the ASHAs. They are trained for screening for malaria by RDT and also to give treatment for the species that is present in here, the infection. For PV and PF, they are trained to give treatment also, so that people are accessed for treatment and for diagnosis as early as possible. Our Principal Secretary, Sir Sampat Kumar, is not here with us today, but he has a very huge contribution in this part. The well-known Tura model, which he has initiated, he was the man behind this model of starting to train the ASHAs at the grassroots level who can diagnose malaria and also give treatment in the sense early and complete treatment. And even like we have started training the ASHAs to give them directly observed treatment. Whoever is detected of malaria, they are the ASHAs, they monitor them while they are still on treatment to ensure that they complete the treatment so that it reduces the mortality due to malaria. So our program, in line with the National Strategy for Malaria Elimination, it is broadly classified into two categories, this intervention, that the early case detection and treatment, and second is the integrated vector management. So in this integrated vector management, we have already discussed distribution of LLINs already being done, usage of LLINs being surveyed, and we have seen that 99% of the communities are using the LLINs. Then we have adult mosquito control, larval control. We all know IRS, it's not very successful because the acceptance by the community is very low. Still, we continue with IRS where the API is more than 1. It's supposed to be 2, but then the API sometimes it depends on the surveillance. So we take in those endemic districts, API more than 1, we spray. So in spite of all these reductions, in spite of declining in malaria, we still have got threats which will lead to resurgence of malaria if we are not alert. So the possible threats, the factors we all know, we know the risk factors that leads to malaria, the socio-economic condition, difficult terrain, inaccessibility, human mobility, we have already discussed. And we have still certain sections of the society who have strong superstitious beliefs in local traditional healers. They are still a choice for many in rural settings, local traditional healers. And with the recent pandemic, the health-seeking behavior of the people has come down. So when we are coming to giving our best efforts in malaria elimination, I think we should go hand in hand. We not only our program we can achieve in bringing malaria elimination to our state as a whole, to the country, but I think now due to declining transmission, detection by microscopy and by RDT may not be sensitive anymore. Like we have seen, many asymptomatic cases are detected for malaria. So it might result in underestimation of the true prevalence of the malaria infection. So I think one of the most important strategy, along with this early case detection and complete treatment and integrated vector management, we have already have some interventions like capacity building and intersectoral convergence with the public and the private, and also with the other sectors. So hand in hand, we should go with the research works and studies. So I'm very happy that you all people are here with us, Shillong IIPH, who is also our next door neighbor. So whenever we need each other's help, please feel free to come to us. We'll try our best to help you all also. And in our need also, we might just come to ask for your help in case we need, because as such we don't have any PCR from our program. So we may need in some cases where we have difficulties in detecting, in confirming the cases of malaria. So I thank you all once again for calling us and for having this stakeholders dissemination meeting. I feel that I'm again back in my MPH days when I was doing after every study, we used to have this type of meeting and discussion with the stakeholders and with our faculty members, with the MPH scholars. So thank you once again, ma'am, and thank you all.
Thank you, ma'am. Thank you for your insights and your continuous support to us. Now we have a few minutes for the question and answer session. So if there are any other questions for the team, or any other general discussion? If not, okay. If not, so we have come to the end of the program. I would like to call upon my colleague, Mr. Peter J. Marbanyang, to propose the vote of thanks to us.
A very good afternoon to you one and all, dear colleagues, dignitaries, and everyone. My name is Peter J. Marbanyang, a Senior Research Officer at the CSCMI here at IIPHS Shillong. I am grateful for this opportunity to deliver the vote of thanks this morning. I hope our esteemed audience found this meeting engaging and fruitful. Firstly, on behalf of the CSCMI team, I thank everyone for their presence here today. I would like to express our gratitude to Dr. Biba Marak, the State Program Officer, NVDSP, Dr. Marba Warshong, the District Malaria Officer, East Castle District, Dr. M. Solia, a Medical Officer, NVBDSP, the State and District Consultant, NVBDSP, and Dr. E. Shadab, Nodal Officer, COVID, for being here with us and for their endless support. I would also like to express my gratitude to Professor Sandra Albert, the Director of our Institute. Ma'am, your enthusiasm, guidance, support, and unceasing ideas have encouraged us all to put in the best effort in our work. Thank you, ma'am. Furthermore, our sincerest gratitude to all the speakers for the presentations, without which this meeting would have not been possible nor fruitful. Last but not the least, I would like to thank and convey my heartiest thanks to all my colleagues whose zealous work in the background would not have been go unnoticed. Once again, a warm thanks to all present here. Thank you. Please join us for tea next door.
Just a reminder, we have a team with the field equipment, so please feel free to interact with them. And can we have our guests to join us the next door for the tea and snacks? And for everyone, tea and snacks are available outside. Thank you.